Mosquitoes are small, almost invisible, but they are responsible for some of the most widespread diseases in the world. In Thailand, mosquito-borne diseases remain a serious public health concern. In 2023 alone, more than 102,000 dengue cases and 98 deaths were reported nationwide. Mosquitoes also spread diseases such as malaria and Zika, affecting thousands of people every year. Yet mosquito populations do not appear randomly. They change depending on season, environment, and human activity. Understanding these patterns is essential for preventing disease. Hello everyone. Today I will be presenting our research titled Seasonal Variation in Mosquito Abundance, a comparative study of Prachuap Khiri Khan and Trang temple sites. Our study investigates how mosquito populations change across different environments and seasons. Why this study matters. Mosquitoes act as vectors, meaning they transmit diseases between humans. Many of these diseases are spread by Aedes mosquitoes, which commonly breed in small containers of stagnant water. These containers can be found almost everywhere. Flower pots, water jars, drainage areas, and temple environments. This raises an important question. How do seasonal conditions influence mosquito populations in these environments? Research objective. Our study had three main objectives. First, to compare mosquito abundance between dry season site in Prachuap Khiri Khan and a wet season temple site in Trang. Second, to identify and compare the mosquito species present in both locations. And third, to assess the container index, which measures the proportions of water containers that contain mosquito larvae. Study locations. Our study examined two different locations in Thailand. The first site was located near Manao Bay in Prachuap Khiri Khan, representing the dry season environment. The second site was located at a temple community in Trang, representing the wet season environment. Data from the Trang temple site was obtained as secondary data downloaded from the GLOBE program database, which provides global environmental monitoring data collected by students and researchers. Methodology. To collect mosquito data, we examine water holding containers that could serve as mosquito breeding sites. These included earthenware jars and containers. For each container, we recorded environmental conditions, including water temperature, pH level, and the presence of a mosquito larvae. Larvae were collected using small nets and placed into plastic cups for counting and analysis. Later, they were examined under a microscope in order to identify the mosquito species present. Key finding number 1. Mosquito abundance. Our first major finding was the difference in mosquito abundance between two sites. The wet season temple site showed a much higher larval abundance. The average number of mosquito larvae per container reached 20.87 larvae per container, significantly higher than the dry season site. A Mann-Whitney test was conducted to compare the two groups. The result was U equal to 127.5 with a p-value of 0.0286, confirming that the difference between the two sites was statistically significant. This indicates that seasonal environmental conditions strongly influence mosquito breeding activity. Key finding number 2. Species composition. We also observed a major shift in mosquito species. between seasons. During the dry season, the mosquito population was dominated by Culex species, which accounted for 93.2% of the specimens. However, during the wet season, the species composition changed dramatically. The population became dominated by Aedes mosquitoes, with Aedes albopictus accounting for 67.8% and Aedes aegypti accounting for 31.3%. Together, Aedes species increased from only 5.8% in the dry season, 99.1% in the wet season. This is important because these species are known vectors of the dengue fevers. Key finding number 3, container index. Another important measurement in our study was the container index, which represents the percentage of water containers that contain mosquito larvae. At the dry season site, the container index was 14.2%. At the wet season temple site, the container index increased dramatically to 65.22%. This means that nearly 2/3 of the containers at the wet season site were active mosquito breeding habitats. A chi-square test was conducted to compare the proportion of infested containers between the two sites. The test produced a p-value lower than 0.01, confirming that the difference was statistically significant. Implications. These results highlight the strong relationship between seasonal environmental conditions and mosquito breeding activity. During the wet season, the combination of rainfall and water-holding containers creates ideal breeding conditions. This suggests that mosquito control strategies should focus on removing breeding containers, monitoring high-risk environments, and increasing control efforts during the early wet season. Community participation is also essential. Working with local communities, including temples and residents, can help reduce mosquito breeding sites and improve public health outcomes.
In conclusion, our study demonstrates the mosquito population significantly vary between seasonal and environmental aspects. In the wet Trang temple site, we found a higher mosquito abundance and a higher mosquito containment infestation rate, and a dramatic increase in dengue fever vector species. Understanding these, we highlight the importance of a data-driven and seasonal adaptive mosquito control environment. Thank you for listening.